Good night, everybody. I'm Jerelle, and I am going to be talking about, I kind of adjusted my original title from what it was before, so that it can like show a little bit more of what I'm going to be talking about instead of what it was previously. So I'm going to be talking about atomistic and locally rigidified energy landscapes for protein folding, and hopefully I don't lose you throughout the talk. Don't get you like too bored. Okay, so what I'm interested in is I am interested in predicting the native structure of proteins. And in my group, we do this using computer simulations and modeling. And the approach that we take is we take an energy landscape perspective. So if you were to imagine every protein has an energy landscape, at the top of the energy landscape, you would have an unfolded state, where you have the fully extended or states that are not folded. And as you go down the landscape, you eventually reach the folded state. Now, sometimes the protein might fall into a metastable state, that is a state that is separated from the folded state by a high barrier, and it might stay there for some time, and that might increase the overall folding time. So you basically have a landscape where you have different structures that the protein can take, and you try to sample how do you get from the unfolded ensemble to the folded one. So this is the approach that we take if we're trying to predict protein structure. Now, the amazing thing about that is that if you're able to correctly predict what that landscape is like for a protein, not only can you predict what its native state is, but you can also predict like the kinetics, like how fast it takes for it to go from the unfolded to the folded state. You could also extract the mechanism, how it actually does it, like which step happens first, next, and so on, and other thermodynamic properties of interest. So now the problem with that, any computer simulation modeling technique, especially when it comes to protein, because for computational chemists, the protein is very big. Like, to the everyday average person, protein is a very small thing, but to us it's like extremely big, and we want to be able to model very large biologically relevant systems, but then that means that is a lot of computer processing time, and therefore we want to develop coarse-grained models by which we can still model these systems, but on a faster time. So we want to be able to get our results relatively quickly, as opposed to waiting for months and months for long simulations to take place. So we're interested in developing more coarse-grained models. Now, if you take the protein and you represent all the atoms and you try and model it, that would be a fully atomistic simulation. So you representing all the atoms in the protein, and that in itself is a coarse-grained model, because you're not really looking at the electronic interactions, you're not getting down to the quantum mechanics, you're more on an atomic level, but we still want to coarse-grain further. So this is where my work comes in, where we use what are called local rigid bodies. So basically, you take sets of atoms in the protein and you classify them as rigid bodies. And now these rigid bodies, no matter how big they are, they only have 6 degrees of freedom. So you have 3 degrees of freedom for your translation and 3 degrees of freedom for your orientation in space. Now you could see that as the protein gets very big and you classify more and more atoms as rigid bodies, you are going to decrease the number of degrees of freedom in your system, and therefore you're going to increase your computational efficiency. But you cannot just take like arbitrary atoms and group them as rigid bodies. There has to be some sort of intrinsic rigidity that you are looking at. So we know that proteins, by as molecules, they do have an intrinsic rigidity, in that if you look at the aromatic rings, if you were to do a very long simulation and follow how the atoms move in aromatic rings, and you average that movement over the entire simulation, there might not be very much motion from the equilibrium position. Also, if you look at sp2 centers and you look at, that is, I just gave this picture for people who are not like very chemistry oriented. So if you look at the centers, the angles don't really change, and you find that they like to remain sort of planar, right? And then if you look at the peptide bond angles, they vary very little for many proteins, right? So if we can explore that intrinsic rigidity and put that into our computations. Now, there have been work that has been done on this before, in terms of some members of my group, what they were trying to find, they were just trying to find the native state. So you have a protein and you're trying to find the folded state, and you define, let's say, the peptide bonds, the rings in the protein as rigid bodies, and then you do the simulation and you try and find the folded state. But what I am interested is, I'm not interested in only finding the folded state, but I am interested in finding out everything you can know about the entire folding process. Now, if you want to know the folding process, you need to also look at transition states, because if you go in from one state to another, you have to transition. So you have to go up barriers to get from one state to the next. Like if you look at the very first diagram, like that would be a barrier, and there would be like a transition state sitting on that barrier. So you want to find out what these are like. And you also want to understand, how does this entire landscape transform when we take rigid bodies, because we know that folding process is a very cooperative process. It's not just something that just happens, boom, bam. You have states where you have like a two-state model where the protein is unfolded and then folds, but generally is an entire process. And when you start saying, okay, this is rigid, it might not necessarily be rigid, and it might move in a certain way that might alter the entire trajectory of the protein. And therefore you want to make sure that you understand how you are changing the entire landscape when you start rigidifying things. And so basically the setup of I know like some of you might be like experimentalists, and you might say like, we do like a real like science, like we actually do experiments and you just do modeling. So I wanted to like put this in terms of like an investigation. So I'm just gonna be like a mock experimentalist for like 2 minutes, literally 2 minutes. So you basically have your control. So my control in my experiment would be the fully atomistic landscape. So I take a test system and I produce the fully atomistic landscape for that, and then I start rigidifying parts of the protein and see how the landscape changes. So my reference would be my atomistic energy landscape. And then my test system, and don't laugh, those of you who've done proteins before, like, this is still so small. Yes, it's actually a very small peptide, only 12 amino acids, but this has been used in a lot of benchmarking for different methods dealing with protein folding. We have to start very small, see what is happening. If we can understand that, then we go to like bigger systems. And then we use a force field to modulate the potential energy and the forces on our system, and we use an implicit solvent to try and pretend like we have like some water molecules in our simulation, which like we just, you know, pretend experimentalists, right? So what we really are doing when we talk about trying to get an energy landscape is we're doing path sampling. So in my group, we basically start from a definition of the folded state, a definition of the unfolded state, and we try and find pathways that connect them. So a pathway would have several minima along the pathway and transition states. So we try and sample as much pathways as we can, and then the sum over these paths using different modulations will help us extract our rates and so on, our mechanisms. So as I said, a discrete path is a sequence of local minima on the potential energy surface and the transition states that connect them. And as we sample these paths, what we're really doing is we're forming a kinetic transition network where we have like a database, and in our database we have the different minima that we find as well as the different transition states. So you have a database with many different structures, and you also have information on how those structures are connected to each other. So if you know that now, you can start thinking of rates, and you can start thinking of mechanisms and so on. So how do we like visualize like what the connectivity of our kinetic transition network is? We use an approach called where we use what is called a disconnectivity graph, and I don't know why they call it a disconnectivity graph since it shows the connectivity, but you know, like scientists just like to, you know, you do it, so like, we like to complicate things unnecessarily, right? So we have disconnectivity graphs which shows us, so each line on the graph would represent a minima in our kinetic transition network, in our database, and then how high you have to go up to get to the center will give you an idea of the barriers between neighboring minima, if you follow me. And then you can see that the unfolded ensemble would be higher up, and then as you come down, you get to the folded state, which is what you had originally defined as your folded state, right? And the interesting thing of that is that for protein folding, these energy landscapes have a very distinctive shape. They are funnel shape. So you have a very broad top, and as you come down, and like, so what is really happening is that the conformational entropy is decreasing. So there are less and less states that the protein can be in as its energy gets very low, right? So now from this initial path, initial transition network, we can extract the fastest folding path. So we can extract the fastest path that will get us from the unfolded state, what we define as the unfolded state, to the folded state. So my unfolded state will be here, my folded state. It depends on what you call your A and your B. The path will be the same way if it starts from on top here and come down to the other side. But I like to do it in reverse because, like, I don't know. I just want to, right? So you go from the unfolded state to the folded state, and then you observe what that path looks like in terms of the barrier heights and so on. And essentially what you want to do is you want to optimize these barrier heights and optimize these distances, because the initial connections that you get might not be the best connections that are there. It's like this analogy when you like think of like well, Google Maps would be like better than us, but like when you try and find like a pathway to get to somewhere, and they give you like different paths that you can get, and some are like 5 minutes and some are like 12, right? So, but we're not as good as Google Maps. We'll find like we might find the slowest path first, and then we might have to keep searching to find like better ones and so on. So essentially what you want to do is you want to grow your kinetic transition network so that you can get better connections and better paths. And you keep growing and you keep sampling, and then you come up with a more connected network, right? So this is the picture of the potential energy landscape that I would have gotten from my model protein trypsin. And from the potential energy landscape, you can then calculate what your free energy surface looks like. And if your computations are fairly accurate, there should be a good relationship between what these landscapes look like. But on the free energy landscape, what changes the most is that you are computing the entropy of the state as well. So the potential energy landscape only deals with what that potential energy is. It doesn't take into account that some states might have higher entropy than others. But when we compute the free energy landscape, you take into account the entropy, so therefore you have like reordering of states and so on. So you might see like an extra funnel appear that was not there before. And then now, since I remember the main goal is for me to look at what my locally rigidified energy landscapes are. So then I need to have some way of extracting information from my atomistic energy landscape so that I can create the ones with the local rigid bodies. And a way of doing that is that you could look at the probability of folding for different states. So you could compute the commuter probabilities, which tells you if I am at a particular state, how probable it is for me to get to the folded state. And that can give you a separation of the different minima that you have in your network, whereby some minima might reach the folded state very easily, have a high folding probability, whereas some might take a very long time to get there. So what I'm trying to do is I am basically trying to understand what are the states that are on the atomistic landscape. I want to know what states are important. So when I make my locally rigidified landscapes, I can see if those states disappear, if they vanish, if local rigidification is changing my landscape in some way, right? And you could also use some structural order parameters. So let's say you could measure like how tightly the protein is folded for all the minima in your landscape. And you could also measure what are the orientation of the rings for all the minima in your landscape, and give you an idea of where do I have like proteins that are very tightly folded, where do I have proteins that have like the rings on either side of my protein, and so on. And so that you know when you do the rigidified landscape, you know what to look for and where to expect states emerging so that you could see what is changing. So this is what we did for TripZip. And once you are able to do that, then that helps you to identify states on your potential energy landscape that may be important. Now, in terms of thermodynamic states, now when we do the path sampling, we sample at room temperature. But we know that we're dealing with matter, and the behavior of matter changes at different temperatures. So therefore, what we also want to do is we want to also look at the states, because when we're thinking of free energy, a free energy is computed at a particular energy. At temperature. Here my temperature is measured in terms of energy, but at a particular temperature is where you measure a free energy, and it changes with the temperature. So you want to see how does the important states on your landscape change as you change the temperature. That is, how does the minima change? So we want to identify these states and also look for them when we are sampling for our local rigidified landscapes. So once we do that, we say that these are the states that are important. This one is in red because I have not sampled it yet. So I put it in red. I'm still like working on connecting it back in, so it's not in the results that I'm going to show you. But we identify the states that are important on our atomistic landscape, and we say, hey, now if I take those states now and I rigidify them and I try and connect them and reproduce my energy landscape, will there still be important states? Will they completely disappear? Because remember, when you freezing out degrees of freedom, you basically limiting how the protein can move. So something that might be stable might not be unstable and might not be a minima anymore. It might vanish completely from your kinetic transition network. So if these ones here I gauge as important, let me see when I rigidify things, are they still important, and so on. And so these are the different schemes that we use. So scheme 1 is where we only rigidify the tryptophan rings. So you have the rings. I don't think this diagram shows it very well. So you have the rings. If you look at the rings in the different structure, although the orientation is different, you see that kind of planar arrangement. So we rigidify the rings, we rigidify the peptide bonds, so trip and pep, and then we rigidify the termini, so the terminal residue. This doesn't really, do much. Let's rigidify it. And we rigidify the tri would be like our trigonal planar centers. So these are the different schemes that we have. So what I want to do is I want to do a progressive rigidification to see where the model breaks down, to see if it breaks down, if it actually works at all. And so then So again, the same picture for my fully atomistic landscape, and then I progressively rigidify and I try and locate what are the states of my system. Now, this database they all have different amount of minima in them. You cannot like really force the system to have like the same number of minima. Is what it finds, right? So they have different amounts of minima. But what we're doing is we're following what states are present on the landscape, what states disappear, and how it transforms. Can anyone like tell me what they think is happening as I rigidify more? Any guess? Just in terms of observation, in terms of like what is in front of you.
It's more orange bottom left.
It's more orange bottom left. Yeah. That's a good one.
It looks like the main funnel is getting sort of steeper, but at the expense of creating sort of other funnels elsewhere where the protein codes.
That is so, you guys are so, oh my god, I love science people, it's crazy. Yes, so what you find is that as you rigidify more, you find that the folding funnel actually becomes very narrow, but the unfolded funnel broadens, right? And you find that a lot of these states that were low in energy, they're now high energy because you are reducing degrees of freedom. Yes, that's all well and good, but you also you're reducing the flexibility on your landscape. So whereas I could take one step and get me from here to here to here, I might be taking 1,000,000 steps and not get out, because this transition state and transition states are very sensitive to changes in structure. Just change the structure by a little. It's like how my supervisor explains it. Is like he say you're on a knife's edge. So basically, one little step and you just completely lose the transition state. So this transition state that could take me from A to B is no longer there because I rigidify itself, so it disappears. So now I might have to go and find 10 of them to get from A to B. So it's like you kind of gain in how much you in your computational efficiency, but at the same time you decrease in the flexibility on your landscape as you rigidify more and more, right? But the important thing is that the states which formed misfolded states on these are the ones in the side funnel. The states which form misfolded states on my potential energy landscape, though their energies are different now, but they still are important misfolded states on my rigidified landscapes. And that is important that we do not lose these funnels even with rigidification. When we get to the very rigidified system, that is when we start to like really push things start to disappear and get crazy. And we kind of want that to happen because we kind of want to see where the model breaks down, because we want to see what is the limit, how far we can push it. And for a very small peptide that we're looking at, it pushed very far before you start seeing things break it down. So what I'm at the stage now is trying to understand all this and trying to rationalize all this and trying to say that are we getting this kind of picture because we use the same treatment for all the systems? Because with rigid bodies, you can alter your set of parameters so that you can sample the landscape better. But I use the same parameters for all of them because I just wanted to see what the picture was. So we're at the stage now where we're trying to figure out, is it because of the treatment that we use, or is it a general picture in terms of rigidification? That as you rigidify more, you sample less of the folded states and you sample more of the unfolded states. And you could also look at how does your fastest path change. So how does your fastest folded path change? So you had the initial path that I showed you, which was optimized, and then so you could say, are the other events changing? Like, did I have like a hydrophobic collapse and a hydrogen bond forming on my atomistic landscape? And now that I rigidify things, is that my protein just going haywire and like the mechanism doesn't really make sense? Like this is pointless, we shouldn't have rigidified the rings, that doesn't make sense. So we want to see how it changes the mechanism as well. So we look and see how that evolves, and what we notice is that if you were to take apart the different interactions that are happening when we do mechanisms, most of these little steps, they might be doing like the same thing. So it might be like a little bond forming, the steps that are very important is where you have your very big jumps. So that means that probably a hydrogen bond formed. And these are the steps that I'm following to see when do they occur on the different landscape. And what I find is that for the moderately rigidified systems, there's very good agreement. Also, for the very rigidified system, there is very, very good agreement. But what the difference is that you find that you go back and forth a lot in the very rigidified system. And that plays back into the fact that we reduce the flexibility. So it might make a bond, and then break the bond, and then make the bond again, and then break the bond, but still in the same area, and then make the other bond, and then still stay there. So you find that the entire path gets longer, and that goes with the idea that we are sampling the unfolded state very long. A lot of our proteins stay in the unfolded region for a very long time before they get to the folded state, and that means that no, that will transfer in the type of rates that we get. So we will see that there will be like a slower rate for these paths as opposed to these ones. So we have to appreciate that that is a possibility, that this kind of back and forth movement is happening on the very rigidified systems, and look for that when we are trying to optimize the folding of proteins on energy landscapes. So yeah, I will not go into that. That's a long detail. So what I want to do is, for my future work, is I want to look at the entire, why I didn't look at this heat capacity, because this was supposed to be like my supplementary stuff, because remember I told you that I did not sample that extra thermodynamic state. So I want to make sure that I sample it first before I give any conclusive discussion on the heat capacity, because heat capacity varies with what is in your transition network. So you can put in just one state and your entire heat capacity curve changes. So I don't want to like make any like solid conclusions on this at this stage. But I want to do benchmarking for larger systems, continue to understand what is happening for my rigidified landscape, and the ultimate goal is to be able to propose reliable methods by which we can use. The fact of the matter is that we know if we use local rigid bodies, our computation efficiency is going to go up. We know that. That's not the point here. The point here is that we want to understand, yes, we know that we can do things faster, but is faster always better? And that is what, if we can determine that, yes, it is actually better, then that can help us to propose ways in which we can implement it into studying protein folding. I want to thank the Gates Cambridge Trust for funding. I would not be able to afford this degree out of my own pocket, never, not until never. And my supervisor and a lot of members of my groups, you do not know these people, but I want to thank Churchill College. Churchill College has been like the bomb. Like, I love my college, and I don't know, I think some people know, but I was like in a cast for like half of last year, and like the college was like so supportive and amazing, and like I just love my college. So yeah, Churchill College, and friends and family. And I want to thank you for coming and listening to me like rant around about proteins and landscapes. Thank you.